East China University of Technology and Science and his PhD in bioengineering from the University of Missouri, Columbia. His PhD work focused on neuroengineering for treatment of neurovascular dementia and Alzheimer's disease. Following that, he was trained at University of Rochester Medical Center on regenerative medicine for vascular applications. He became an assistant professor in 2010, where he focused on novel biodegradable metallic materials for tissue engineering and regeneration. In 2016, he moved to Texas as an associate professor to continue his research in biomaterials and regenerative medicine. In fall 2019, he was recruited to Stony Brook and currently has a research interest in biomaterials, cardiovascular diseases, musculoskeletal disorders, as well as aging and neuroscience. He has received more than 10 million extramural research funding and authored more than 100 papers. He's an elected fellow of American Heart Association. Dr. Zhu also serves as editorial board of many scientific journals, as well as grant review panels, including NIH, NSF, FDA, and NASA. The floor is yours, sir.
All right. Thank you very much, Magmaris, for the nice introduction. Let me thank you, Ali, and all colleagues for this nice invitation. And it's really my honor to give a talk at your seminar series at Terasaki Institute. So today, my topic is recent advances in bioresorbable or biodegradable metals as medical implants. I will mainly focus on the most popular application, including cardiovascular stent and orthopedic bone application. So my part one would focus on the evolution of bioresorbable metallic stent over the years. And the second part, I'm going to talk later on about 3D printed or additive manufactured magnesium-based bone scaffold. So for stent, there are several types, including the bare metal, drug-eluting, bioresorbable, permanent, or surface modified, or even some self-expanding stent. So all these kinds of stent have their own pros and cons, I guess. For example, showing in A is some neointima hyperplasia response from a bare metal stent. And in B is the illustration of protrusions, malpositioning, and later stenosis of even for some drug-eluting stent. And for C is the showing the better vascular healing from implantation of a bioresorbable stent. This is largely due to its disappearance later on. So no more implant, eliminating the potential clinical complications associated with the implant. And there's also some versatile response of surface modified stent as showing in D. And lastly is the self-expanding stent, which is more suitable for peripheral or cerebral vascular instead of cardiovascular area, thanks to their higher tortuous paths and pulsatile forces. From a clinical perspective, the bare metal stent often result in restenosis, and the metallic ions released over time may introduce some adverse effect, like the hypersensitivity, the stent fracture. Also, the example here is a multi-link vision stent associated with hypersensitivity to metal ions. Whereas for bioresorbable stent, vessel is healing while the implanted material is gradually disappearing. And here is some in vivo histology and OCT images of a polymer-based so-called ART BRS stent. And some micro-CT imaging also showed a 1 and 20 month implantation time of a magnesium stent. And lastly is a micro-CT imaging of a pure zinc stent implants over a 12-month period in rabbit. Both permanent and bioresorbable stent can have drug-eluting features. So on the panel A is some SEM image obtained in vivo of a PM EDS, which highlights the EES coating over its competitors. And in B, it is a sort of demonstration at the molecular level, illustrating that even DES may also impair the re-endocellularization of the stent material, which could result in strut malposition with some consequence of late or very late on stenosis. For the bioresorbable stent, on the left is some OCT image showing the high percentage of embedded with lower percentage of protruding and the percentage of uncovered struts of a magnesium stent. So this is the magnitude BRS and the absorb BVS. Both are the magnesium-based stent. Of course, there are other type of examples, including the lately the iron-based and some nitride-coated iron-based EDS. And other example is magnesium-based with PDLA carrying rapamycin release coating. So these are the sort of new development in the stent field. For metallic stent, traditionally they are non-degradable, mainly stainless steel, and the representative one is, for example, the R stent from Auber snake. There's another popular is cobalt chromium. One example is the Vision series from ABOT, and also platinum chromium alloy, the typical one like the Omega BMS from Boston Scientific. So these are all the conventional stent, but they are non-degradable and also already used in clinical. However, for biodegradable, three major materials are magnesium, iron, and zinc. For magnesium one, they already got CE mark in Europe. For iron and zinc, largely still under development or clinical trials. Here's some comparison of the mechanical properties of different metal materials. We look at the traditional stainless steel, the bioresorbable magnesium and zinc. So the stainless steel obviously have superior tensile, fatigue, and creep strength compared to magnesium and zinc. On the other hand, the magnesium and the zinc have similar tensile or fatigue strength. And magnesium have a little higher creep strength than the zinc. The elongation rates of the zinc showed a discrete distribution with average value in between stainless steel and magnesium. Overall, all three materials can meet the minimal mechanical strength as a stent material, although magnesium zinc may need some prop alloying strategy or some mechanical thermal deformation or treatment to further enhance their mechanical property. Also here showed the mechanical stability is characterized with the percentile of retained strength over the degradation time. The inert stainless can keep their strength stability during the whole implantation period, while the zinc material have little decrease on their strength after 2 weeks of degradation, and they have about 80% of strength after 8 weeks. The magnesium lost around 20% and 35% of their strength in 2 and 8 weeks, respectively. Lately, there are new zinc alloys were developed, so they showed superior mechanical properties. If you look at these two figures, especially the zinc lithium alloys or the zinc lithium magnesium alloys, their mechanical property can reach or as high as comparable to the medical grade titanium. So this is sort of opens the new window for using magnesium, potentially to replace some titanium-based implants later on, providing the very good mechanical property in the zinc and lithium or zinc lithium manganese alloys. Also, another good thing about magnesium and zinc material is their antibacterial property. So for example, here for stainless steel, both E. coli and S. aureus can form intense biofilm, while on the magnesium and zinc surface, you see significantly less attachment of those bacterial strain. And their antibacterial rate is about from 40 to 60 percentile in the solution, and on the material surface, it's almost to 100% antibacterial rate. And the magnesium stent is already approved in Europe. Their trademark is Magmaris, so-called, the latest one is the second generation of dreams. So from this, they are made of WE43 with drug-eluting feature. And it's the data, the OCT data in human trial shows that after the PCI operation, there is a high attenuation value, and it indicates the scaffold strut over time. And the signal subside of the OCT over time indicates the bioresorption of the struts. So here the star in the figure indicates the calcium component in the tissue due to the degradation of the magnesium material. The arrow is pointing to the stent strut remaining. And this is also in human trial, the ultravascular ultrasound data. So the DREAMS 2G showed a continuous or favorable safety profile and a stable performance up to 12 months in human trials. And following the implantation, the restoration of the vessel geometry, vessel motion, and the bioresorption were observed up to 12 months. And also the lumen size was largely preserved. So the outcome is pretty good. And here is some latest development for the drug-eluting iron stent. So this is the configuration, the structure, and the angiography of a sirolimus eluting iron stent, so-called IBS. This is in pig. And another EES made of cobalt chromium used as the benchmark control. So from the QCA, the quantitative coronary angiographic imaging on the top, and the middle is the OCT imaging. Bottom is the micro CT imaging. These data shows that the drug-eluting iron stent so far is very good, comparable to the benchmark control. And on the right side is the percentage of the area of stenosis. So the ratio of the scaffold area at follow-up. So the S to the initial just post implantation S0 is also comparable between these two stents. So far, the performance of the drug-eluting iron stent is, if not better, even very close to the benchmark control. This is a good sign. But the beauty of these iron stent, they will eventually disappear, not like the control. So this is the biggest advantage. And the histological analysis showing has comparable operability, also the midterm efficacy, safety, all comparable to the cobalt chromium control. And also the IBS showed no scaffold associated thrombosis, no severe neointimal hyperplasia, no also good mechanical support after 180 days. However, the degradation is still minimal for this IBS, largely intact. So it may take a while, probably a few years to completely disappear. Now for another material is zinc-based stent. So here is some study in the rabbit from our lab and with our collaborators. On the left side is the histology. The B is the angiographic imaging showing the implantation of 2 stents in the rabbit. And we observed pretty rapid endothelialization on the material surface. The smooth muscle cell proliferation was low. The stent showed a complete patency of the artery even after 12 months. The diameter vascular vessel stenosis is less than 10%. From the macrophage staining showed no acute inflammation in 3 days after implantation. The inflammatory cells, largely macrophage, increased after 1 month. It largest reached at 30 days, probably largely due to the degradation product after a while. And then a decreased trend of the inflammatory macrophage infiltration was observed after 3 months and gradually back to the baseline after 6 to 12 months. And this is SE imaging showing the endothelium formation on the material surface. After 1 month, the endothelium layer is already pretty good, and a confirmed monolayer formed, showing the elongated endothelium cells nicely cover the stent strut after 6 and 12 months. And also the histological staining showing abundant collagen formation and the fibrous elastin at 6 and 12 months. And the extracellular matrix appeared to be very normal, indicating the good biocompatibility of the zinc stent material. And these are the micro-CT and SEM imaging showing the volume loss of the material after 6 months, about 20%, and about 40% after 12 months. The corrosion behavior is largely uniform after 1 month. However, some localized corrosion were observed at 12 months. And we also did some preliminary study using zinc lithium alloy stent in a pig model. So this is the micro-CT imaging as well as the OCT after 6 months. So the stent looks like still largely intact, and the lumen is remained open shape and pretty normal as well. And here's some preliminary histological staining. The smooth muscle cell proliferation, however, was a little high. So this is some neointima was observed, and some surface degradation of the material as revealed by the SEM imaging. Of course, this is pretty much preliminary. More investigation for a long time period are needed. Lastly, we checked on the systemic toxicity of the implants. So far, we observed no infarction, histological changes, or corrosion product accumulation in the internal organs. All these organs pretty much looks very normal, healthy, histological morphology. No significant difference in the blood level of zinc ion were observed compared to the control. So far, no bigger concerns about the systemic toxicity due to the degradation product released from the zinc implant. So the summary for the stent research, the drug-eluting magnesium, the second generation DREAMS, Magmaris, already showing very promising clinical or human trial, and some magnesium based Bioresorbable stent named Unity B. It's under clinical trial or in the clinical usage in Europe. And all these magnesium-based materials showed very good biocompatibility. The weak mechanical property and relatively fast degradation could be a concern. And the inflammation was low to moderate level. The late or very late stenosis or thrombosis need further investigation. For the drug-eluting iron, the latest is so-called the IBS angel. They showed strong and stable mechanical property, good biocompatibility. The degradation rate was a little slow, and the possible degradation product retention over a long period could be a concern. Inflammation was low to moderate. Same as magnesium, late stenosis or very late thrombosis need further evaluation. For the biometallic zinc, so far in the animal model, it showed a very good degradation rate, quite suitable, not too slow or not too fast, good biocompatibility, some adds-on beneficial feature like antibacterial property, mechanical property after alloying with lithium and manganese was excellent. The inflammation was moderate. There's some moderate to severe neurointimal hyperplasia were observed in the pig model. Of course, the late stenosis or very late stenosis and the thrombosis need further evaluation. So regarding the high level of neurointima, maybe a drug-eluting feature could solve this problem. So we are trying to, at this moment, coat the zinc with different polymers with the drug incorporation combined with the good material property of the zinc base could be possible to develop some nice bioresorbable stent in the near future. Okay, next I'm going to move on to bioresorbable bone scaffold, largely focusing on magnesium. So here is a clinical challenge. Bone defects caused by different reasons like trauma, tumor, infection, usually need some help from implant, especially when the defect size over some critical healing size. And for the bone, from the implant perspective, to tailor a better regeneration, we would like to have porous structure to foster a better healing. If the defect is irregular shape, then 3D printing could offer some advantage. For bone defects, if there's a strong mechanical support is needed, then usually metals are superior than the polymeric counterpart. So that's how additive manufacture or 3D printing approach could show really advantage. So here's some example of 3D printed scaffold currently used for bone application. It's because bone have different shape and property, and the treatment of these bone defects largely need a customized implant. So compared to the traditional bi-inert titanium, the bioresorbable, therefore the magnesium alloy, both of them have their advantages and disadvantages. For titanium, the mechanical strength, bioactive and biocompatibility, excellent osteointegration is a good thing. But the disadvantage could include the revision surgery or second surgery or removal surgery due to long-term inflammation or the stress shielding effect induced loosening of the implant at the interface. For magnesium, greatest advantage is the bioresorbable property, so they can eventually disappear. And the mechanical property is more or closer to that of natural bone, therefore reduce or minimize the stress shielding effect. However, their long-term in vivo degradation behavior and biocompatibility analysis are still needed to warrant a further clinical application. So here we will investigate the in vivo degradation behavior of 3D printed magnesium scaffold, and particularly look at the interaction between the bone and the scaffold. So the material is magnesium We43 alloy, and the benchmark control is a titanium grade 1 medical titanium. we used the laser powder bed fusion approach for 3D printing. Here is the optical imaging showing the titanium and a magnesium cylinder with porous structure. The diameter is about 4 millimeter. Length is also about 4 millimeter. But the pore size is 250 micron. The struct size is about 250 micron. And the compressive modulus of these two material is around 1 to 2, between 1 to 2 gigapascal. Titanium is a little bit higher than magnesium. The implantation model here, we used a rabbit. So 2 cylinder defects created on the femoral bone by drilling 2 holes, and the scaffold were then inserted at 2 positions in the femur and pressed to fit in the defects. And we evaluated the outcome at week 5 and week 25, representing the early stage as well as the later stage of bone regeneration and bone remodeling, respectively. So we used the micro-CT, SEM, EDX to do the characterization of the biodegradation, and using the histological staining, mainly Mason Goldner, for the biocompatibility assay. So we look at the 2 planes, the lateral at the cross section and the longitudinal plane, to look at the scaffold and the bone tissue. So for the cross section, we look at the defect region. Those are embedded in the bone tissue, as well as the region B is the cavity region, so exposed to the marrow cavity of the femur. And from the longitudinal plane, we look at the central region as well as the peripheral region of the implant. So left is the whole volume of the implant, include both the metal and the degradation product, the metal oxide. So about 70% of the original after 5 and 25 weeks. Although the whole volume shows no change, the ratio between the metal and the oxide switched. So the percentile of oxide, magnesium oxide or other oxide, increased. So indicating an ongoing biodegradation process of the implant. Then we look at the 2 regions, A and B, as I mentioned, corresponding to the bone embedded in the femur and the region exposed to the marrow cavity. So the degradation is more aggressive in the cavity B region, suggesting the scaffold in the cavity is absorbed faster. And this is actually quite desirable for our implant. And then here we look at the another plane of the 2 regions, the central versus the peripheral region. So the degradation is more aggressive in the central area. And in general, the degradation is less aggressive in the region C, so where is exposed or embedded in the bone tissue, than the one exposed to the bone marrow cavity. This is probably largely due to a lack of the body fluid or the electrolyte inside the bone tissue compared to the bone marrow. This is 5 weeks after implantation magnesium scaffold. We look at two regions, A and B. A is exposed to the bone defect, while B is exposed to the marrow cavity. The interface is checked by micro-CT and XRD EDS. So at the region A, the oxide layer is relatively homogeneous and relatively thin, while in the cavity region, the degradation behavior is stronger and have a thick or different type degradation product layer, including magnesium oxide, calcium phosphate compound, and also some mineralized bone product. And this is 25 weeks after implantation. So still there are some thin oxide layer covered by mature bone layer at the defect region A, and while some great portion of the scaffold is already converted to oxide and the calcium phosphate compound in the cavity or in the B region. This is a titanium control after 25 weeks. So at the defect region A, a thin layer of calcium phosphate compound was observed on the surface. And in the cavity region B, there are also a calcium phosphate compound layer on the surface, but there are also some mineralized bone is partially connected with the titanium scaffold, indicating a good osteointegration. And then we look at the overall bone regeneration. So the bone volume is significantly higher for magnesium than the titanium, indicating the magnesium is pro-bone regeneration compared to the titanium. For the titanium, there's some new grow bone inside the implant, and they distribute quite homogeneously. For the magnesium scaffold, the new grown bone seems more or preferred at the peripheral region, where is more closer to the natural bone structure. And this is some SEM micro-CT imaging, so showing week 5 and week 25, corresponding to earlier bone regeneration and the late bone remodeling. So compared to the titanium, magnesium provide some additional feature indicating that they might can guide the bone regeneration during the period, and more thick mineralized bone layers were observed in the defect region. Then we look at the histological analysis. This is week 5 for the magnesium scaffold. Vascularization is observed both inside and around the scaffold. Also, some mineralized bone or osteoid is observed inside and around the scaffold. Here is the titanium control. Similarly, vascularization is observed both inside and around the scaffold. Also, some mineralized bone on the surface observed in the defect region. And this is a sham control without any implant. Interestingly, there's some 2 layers of bone are formed at the different position. and vascularization is observed around the outer, the newer grown bone. Also, then the later stage, 25 weeks for the magnesium, compared to the earlier stage, a thick mineralized bone is observed both inside and at the surrounding area of the defect. We also observed some aggregation of cells in the cavity region, as well as the bone structure inside of the bone structure. Those kind of feature is very obvious, and we did not observe such a thing in the titanium implant. So only some mixture of mineralized bone and osteoid in the defect region were observed. Less mineralized bone inside or the scaffold. Some relatively thick mineralization bone is observed in the cavity region, however, but no aggregation of the different blood cells were observed. I guess this is indicating less vascularization for the titanium case. Here is the shame control. Again, the two layers of bone still exist. The outer layer, however, is thicker than the inner layer. Vascularization is also observed around both the inner and outer layer. Some mixture of the mineralized bone and osteoid remain around the outer layer. Lastly, we checked on the systemic toxicity by looking at the histological analysis of different internal organs. So no sign of the systemic toxicity. The hemolysis and lipemia index of the blood are normal, and the chemistry panel from blood analysis shows the different group have no significant difference, and all are within the normal range of these parameters. So again, the systemic toxicity appeared to be not a concern. So here's a summary for the magnesium bone alloys. So about 41% metal is converted into oxide, and around 70% of the whole volume remains even after 24 weeks. However, the ratio between the degradation product and the metal change with the oxides increasing over time. Magnesium degradation behavior is not homogeneous for the whole scaffold. It's more aggressive in the peripheral region. And the distribution of the new grown bone inside the magnesium scaffold looks like it's stronger, or it prefers the peripheral region, while for the titanium is more homogeneous. And there's a formation of some compact mineralized bone layer in the defect region for the magnesium, while it's more often at the cavity region for the titanium scaffold. And the magnesium scaffold looks showing a better guidance for the new bone regeneration than the titanium alloy. Okay, so this slides showing the research interest in our lab. We are mostly working on biomaterial medical device by 3D printing using traditional and bioresorbable metals and other materials. Another research interest is neuroscience and neuroengineering. we largely focusing on aging, dementia, and Alzheimer's disease, using a different approach, including the artificial or the in vitro brain organoid approach. This is the overall picture of our research. Here is some ongoing project. Largely are the bioresorbable metallic implants, 3D printed for bone scaffold, stent, and surgical device. Also, bioresorbable polymers and hydrogels for wound healing. Lately, we are also trying to using DNA orgamide or hydrogel for tissue engineering and drug delivery. Nanoparticles, especially nanodiamond for treatment of osteoarthritis. And Alzheimer's, we are focusing on a role of bimetals in neurovascular, BBB function, the RAGE role and AGEs, and sex differences, and etc. Lastly, I would like to thank the funding agency and our group members and wonderful collaborators. It's really fortunate to have them to work together on all these research projects. Thank you very much. That's it.
Thank you for this fantastic talk, Professor Dansu. A couple questions came in. So, is there any side effect if large amount of zinc or magnesium degraded in the body?
Sorry, let me turn down the volume a little bit. Can you repeat the question?
Sure. Is there any side effects if large amount of zinc or magnesium degrades in the body?
This, I would say, the adverse effect would be minimal, but of course it's always a concern if they cannot be cleared by the body in a harmless way or in a prompt way. So far, but the good thing is that both magnesium and zinc are, sort of essential element. Okay, but everything has, toxicity, especially when it's overdosed. So if over a long-term period that the level of the magnesium and zinc or even iron is exceeding the maximum limit, of course, it will be toxic. But if, as long as it can be quickly secreted by the bloodstream through urine or the liver or by the kidney, the toxicity is so far is not a concern according to our data. But of course, a long-term evaluation might be needed to play it safe.
Thank you. Does the similarity among calcium and magnesium ions post-degradation have facilitated mineralization? Do you have XRD data?
Yes, probably we didn't show it here very a lot of data here due to the limitation, time limitation. Yes, we are working on these analysis, especially at the interface using all kinds of techniques, including the XRD.
Thank you for that. Could nanosizing of the grains in bioresorbable metals be leveraged for lowering the alloying additives or as an alternative for alloying, regarding the increased grain boundaries which encourages them?
Yeah, definitely. These metals usually needed to be alloyed with some other elements to greatly reduce the grain size or enhance the mechanical property, also may change the degradation product a little bit. But when we have to be careful by selecting the most suitable or the most prop elements. Because of a toxicity concern, we want to use some safe element to avoid some other element like rare earth or something. So this is one consideration. Second is the amount of the other alloying element. So whenever we talk about toxicity, it's always related to dose, right? So the less amount, I guess, is better. So far for zinc material, we and others, our collaborators, all discovered that adding lithium could significantly, so it's like a huge jump to increase the mechanical property of zinc alloys. But sometimes it's at the cost of other mechanical property, especially the ductility. So that's why we are trying to add additional alloying material, alloying elements like magnesium or manganese to reach a balance between Young's modulus or tensile stress versus ductility. So far, it looks promising.
Thank you. Well, that's all the questions I have. I just want to thank you for your time. I appreciate,
No problem. Pleasure is all mine. Thank you again for this wonderful opportunity.
Thank you. Have a nice day.